Okay, you guys, this is Unit 6, Segment 3, Protein Synthesis. And this image is kind of showing you the overall overview of what's going on. So we have DNA, double-stranded, in the nucleus, and then RNA is being made. RNA can leave, comes out, finds the ribosome, and then new RNA nucleotides will bring in, tRNA nucleotides will bring in proteins, which ends up to be a long chain. So that's ultimately what's happening. Protein synthesis. Synthesis means making, and it's proteins. There's three important points to remember. This is called the central dogma of biology. The chromosomes are made up of DNA. Chunks or segments of the DNA make up genes, and these genes code for a protein. The proteins in return relate to the trait, and the traits can be eye color, enzymes, hormones. Everything about you is your traits. So ultimately it goes from DNA into proteins into traits. You should make a chart because you're going to need to compare DNA to RNA. So on one side, DNA, the sugar for DNA is deoxyribose, but the sugar for RNA is ribose. But look at how similar they look. They both have a phosphate, they both have that sugar, they both have a nitrogen base, but the sugar is different. With DNA, the sugar is deoxyribose. With RNA, it is ribose. Okay, they both are 5-carbon setups, so they both look similar. DNA is double-stranded, RNA is single-stranded. DNA can never leave the nucleus. RNA can leave the nucleus and does. And then the new nitrogen bases, DNA has A, T, C, and G as options. RNA does not have T, but it does have A, C, and G. It replaces the T with uracil, which is very important to remember. Whenever you see a chain that has a U in it, you know it's RNA because DNA doesn't have uracil. So there are some similarities and there's some definite differences. Okay, so the first step that happens in the nucleus, because that's where the DNA is called transcription. You should just say it out loud. Transcription. And then an mRNA, so there's different types of RNA, okay? mRNA is the messenger RNA, and it is actually created inside the nucleus. It's shown in blue when the DNA splits open, okay? So the sides can be read as a template. RNA comes in along and is RNA polymerase unzips and brings in RNA nucleotides to form the new strand of RNA, which is in blue here. Sorry, I can't speak today. That will only be as long, the mRNA strand will only be as long as the gene is. So once it reaches the end of the gene on the DNA, it stops, and that RNA breaks loose and acts like a snake and slithers out of the nucleus. I'm going to be showing you guys some animations in class, so I'm going to skip those for the notes for now. But here's a nice visual. So what do you think this is? It's got openings in it. If you said the nuclear envelope, you are right. This is the outside of the nucleus here. This is your DNA. What do you think this enzyme is? It's unzipping and bringing in new RNA nucleotides. So this is RNA polymerase. Very similar to DNA polymerase, but RNA polymerase makes RNA. DNA polymerase makes DNA. So here's your RNA snake, and it will come out, slither out through that opening. So the making of this RNA strip is called transcription. Translation happens outside the nucleus. And here's a couple things that you should write down for translation. So mRNA, messenger RNA, is read, and amino acids are linked together to form the protein. 3 mRNA nucleotides equals a codon. It's called a codon when you have 3 of them. So if you have A, U, G, 3, a sequence of 3 nitrogen bases, that equals a codon. 3 complementary tRNA nucleotides is called an anticodon. So tRNA, guys, is transfer RNA. So mRNA is what's made in the nucleus. and it copies the code of the DNA. tRNA are little hairpin RNA molecules that are floating around, and they have amino acids attached to them. So they're the ones that are bringing in the amino acids. And they're complementary nucleotides to the codons. They're called anticodons. So if the codon is reading AUG, the anticodon is UAC. And tRNA brings in the amino acids. All right, here's what translation looks like. You've got the snaking mRNA sequence coming out of the nucleus. It comes out, and you've got the recognizable nitrogen bases here. It's read in groups of 3, which are called a codon. So AUG right here, this first 3. And here's the hairpin tRNA I was telling you about. It looks like they say it looks like a hairpin. It's got a little bit more bumps than a hairpin typically has, but underneath here is what's called the anticodon, and this is the complement to this, which is the codon. And this strand right here is the mRNA. This right here is the tRNA, and then tRNA, you already wrote down in your notes, brings in the amino acids. So here's the red dot amino acid, and this happens to be one called lysine. This blue blob is the protein synthesis organelle called the ribosome. So the ribosome just kind of moves along, bringing in tRNA. It locks in, and once this tRNA locks in, it pops off the amino acid and it links to the one before it. So this thing kind of scoots along, adding on amino acids to amino acids to amino acids. So you can kind of see how this is working. tRNA pops in, and the next one pops off, and each time it pops off, it's losing that amino acid to the next one. So you end up building this long chain. Now each little dot is an amino acid that's a monomer for a protein, so the whole long chain is called a protein, but you're also going to see the word polypeptide. And the reason for that is because peptide bonds are bonding the amino acids together, so they call them a polypeptide. Poly means many, and many peptide bonds there. So tRNA leaves, new tRNA comes in, with the anticodon matching with the codon, popping off the amino acid, off it goes. You're going to see this genetic code in your book and in your packets, and it's a way to start to break down what the codon is from the mRNA and how it translates into the amino acid sequence. So if you are given a sequence of AUG, you start from the middle, A, and then you work your way out, U, and then G is here. So the amino acids are along the outer edge, okay? And AUG codes for methionine. So methionine would be the amino acid, the little ball that comes in and gets connected to the next amino acid. We're going to play with this. We're going to play games with this in class, so you'll get more comfortable with it, and you will see it on the test. So how are proteins synthesized from DNA? I'm going to go back through, review. DNA is transcribed, that's a vocabulary word, transcribed into mRNA. mRNA leaves the nucleus, it's able to leave, and DNA can't, and travels to the ribosomes that are floating around in the cytoplasm. The ribosomes in the cytoplasm use the codons on the mRNA to actually translate that into amino acids. So you have transcription and translation mentioned in here. The tRNA hairpin molecules bring the amino acid little balls to the ribosome. The amino acids form a chain. If you guys remember in that one image, it was polypeptide, and those polypeptides are proteins. The proteins, my friends, are what make your traits. So it goes like this: DNA to RNA, RNA to amino acid sequence. Amino acid sequence makes the proteins, and the proteins make the trait. This is another central dogma of biology. I have 2 arrows here. What do you think they represent? Going from DNA to RNA. The answer is transcription. And then going from RNA, in particular mRNA, to the amino acids with the help of the tRNA. This is called, answer is, translation. Okay? So transcription happens in the nucleus, translation happens in the cytoplasm at the ribosome. We're going to do some practice in class, so I'm actually going to hold off on that, and I think we're just about done. So quick review, how does a molecule go from DNA to RNA to amino acid? You need to come up with those steps. I kind of went over it already. So DNA to RNA is transcription. RNA to the amino acid sequence happens with the help of tRNA, and that is translation. Something along the lines of that. All right, I think I am done. So that is segment 3. Lots to do in class, lots of practice. This will not get you everything you need to know. Definitely the practice in your packets will help with that. All right, thanks.